This is a re-upload of my previous video series on geometric morphometric analysis, now combined into one long-form video with a few updates and edits to make it more accessible and useful for your studies and research. If you found any of my content helpful, then please consider liking my videos and subscribing to the channel, as it really supports my work and helps me continue creating educational content. As a self-funded PhD student, I'm always open to collaborations and new opportunities, so feel free to reach out if you'd like to work together or collaborate in projects in the future.
So what is geometric morphometrics? Well, traditional caliper-based measurements only really describe the distance between 2 points. They don't describe the points in relation to all the other points, and they also don't describe the angles between the points, so you can miss a lot of variation between the shapes. So to overcome these problems, a more sophisticated method called geometric morphometric was created. So, relative positions of landmarks are compared between individuals or groups, using the set of landmarks to describe the shape. So a landmark is basically a 2- or 3-dimensional point described by a set of rules. So with a set of rules, it must be present on all the studied organisms, or it has to be marked approximately, or not used at all. The number of landmarks cannot exceed the number of specimen samples, and then the results do depend directly on the quality of the landmarks. There are 3 types of landmark. True landmarks, which have some biological significance, so for example, origin of the dorsal fin there. Pseudo landmarks, which are defined by relative locations, such as the point of highest curvature of this here. And then there are semi-landmarks, which are defined by a location relative to other landmarks, such as midway between landmarks 1 and 8 would be landmark 9. So landmarking is one of the obviously most important parts of geometric morphometrics, but the second most important part is the Procras analysis. So if we were to plot all of these points for all of our samples after doing the landmarking, just on a graph, it would look like this, just a mess basically. So what we do with the Procras analysis is, if you imagine these are 2 sets of landmarks, first all the configuration of landmarks are scaled to the same size, then they are transposed onto the same center of gravity, and then they are rotated to the same orientation, and this allows them to be compared. So if you were to plot Procras coordinates, this is what it would look like. So these are going to be all the sections, this set of videos. So first I'll go through the practical methods, how to photograph the fish, and how you can best avoid error to get the best results you can. Then I'll go through photo editing. This isn't an integral prerequisite of geometric morphometrics, but it can help to pick out the features, particularly because landmark can be quite a boring task when you're going through all of these fish. And also it'll be great to make figures when you do your final paper, because these photographs can really make your graphs pop. And then in the next part, I'll show you how to do all the landmarking. Once you've got your coordinates, there's sort of 2 methods that I'm going to show. First, there's MorphoJ and SPSS. This is the simplest method if you're not really used to using any of these programs. But then the second method is R, which can be quite complicated if you haven't used it before. But if you're doing large amounts of data and you might have to go back and add in other things, you can just repeat the code rather than having to go through all the methods again in the other programs. So this video will cover the methods required to obtain suitable photographs for the geometric morphometrics and how best to reduce error. There are a few important things to remember. Keep everything uniform when photographing specimens. Use the same camera and same methods for each specimen. The more variables you keep the same, the more reliable your results. This is especially true if you end up needing to take additional photographs of the fish or more fish at a later date. If any fish are incredibly warped, it's best to exclude them, as they'll increase the error in your final results. This is caused by something called jar effect, so you might have fish with really bent tails or squished bodies. But sometimes you don't actually have an option, you have to include them, as you might not have enough well-preserved samples of that population. So I'll also show you how to pin them out and get them to look as straight as possible. You also need to find a way to stabilise the fish so it doesn't lean. You can use blue tack or pebbles. I'll be showing you how to use it with blue tack, but in other non-fish based studies, such as skulls and things like that, they often use pebbles more often. And it's also important to use a background such as black or grey, so then we'll be able to adjust the colours later if there was any sort of yellow glow from the lighting. And these are all the equipment that we'll be using. We'll have like a foam board, pins, a coloured background, a scale, which will be really important when we're landmarking later, a camera, tripod, spirit level to ensure that you've levelled the fish well, lighting, so like from the lamps so we can avoid shadows, and then the blue tack stabilising. So why do I keep going on about error? The reason it's so important to use the same camera for every photograph is because the camera lens distorts the images, and the level of distortion depends on the lens, camera, and then other variables such as the zoom and distance between the specimen and the lens. The best way to minimise distortion as much as you can is avoid mixing data sets from different cameras, lenses, or photographic setups. Avoid placing the specimens or the scale bars near the edges of the photographs. Maintain the same camera settings as much as you can for every image. And make sure you use a reference grid or a scale so you can correct for or quantify the distortion. Otherwise, you can have your results completely invalidated and you can't be certain of your conclusions. So, for example, all of these photographs were taken by different photographers, different cameras, lenses, levels of zoom, and the distance from specimen to the lens. If some are angled, the scale bars are sometimes near the edge of the image, so it'd be incorrect to make conclusions from results obtained from these photographs. However, these images, they all have the same level of zoom, same distance from camera, and they would be suitable for comparisons. So a very important step once you set up is to do some initial tests to see which height and level of zoom produce a smallest amount of error and whether flash helps or hinders your efforts. So you can take photographs of the same specimen with different heights and levels of zoom and then measure parts of the fish, both physically and on the computer. So for my camera, I found the best setup was me above the fish with a 5x zoom and no flash, but it depends entirely on the camera, so you should mess about with your settings a bit to find out what works best for you.
So sometimes you'll have a fish like this, where the fins are all nicely pinned out and you can see where they start and begin, where you don't actually need to pin it out. But more often than not, you'll have a fish like this. You can't really see exactly where the fins start and begin. So with a fish like this, you will need to pin it out. So I'll show you how to do that. So put it in the middle. This is foam underneath and then a felt layer so the pins can easily go through. Zoom in there. So to start with, you'll want to secure the felt onto the foam because you don't want that moving around while you're pinning your fish. Then you'll want to secure the fish. And then once we've done that, we can get on with actually pinning our fins out. So for the dorsal, you want to try and find, easiest to use the side of the pin, the start of the dorsal, so the first spine. And then pull it out, so you're splaying all of those. And then it will likely dip in the middle, so you'll want to pull out there as well. And then you'll want to do the same with the anal fin. Quite often you can find that they actually dip behind each other. So I think I can see there that the first one is actually dipping. There. So make sure that you flick them out like that, and there's always going to be sneaky ones. And then you'll want to do the same with the pelvic fin. Finding that first spine, and make sure you don't pull that out too far because it will just look unnatural. So that's basically what you need to do. And now then when you try and do your geometric morphometrics, you'll be able to see exactly where the starts of the fins are. And it'll be a lot more accurate than just trying to guess when they're flat. So now we're going to go through how to obtain the photographs required for the geometric morphometrics and how best to avoid error. If you're working in the laboratory, make sure to wear a lab coat and gloves, and if you've got wild lockdown hair like mine, you tie it back. So, first thing is, once you'll set up your tripod, make sure that the height is the same as the one that you found during initial tests to have the least amount of error. If you have got your fish pinned, like this one, then you're all set. But if you aren't pinning your fish, then you'll need to pick a background. So I've got black card and grey felt. And then get out your fish using forceps. Make sure that you dab the ethanol off it, because you don't want it to be shiny in the photo. If it's particularly moist, then make sure that you perhaps leave it for a little while for the excess ethanol to evaporate. Because also with the card, it can get warped if it gets the ethanol on it. So then you need to place the fish on. You want to make sure that it's straight and not at an angle, so you can use blue tack for that. In your photograph, you're going to need a scale, which we'll use during scaling in the landmarking section. Make sure it's not too close to any of the edges of the image, because it can get distorted. So now you want to set up your lighting. You're going to do this to avoid shadows on the fish, so you can show all the features. So just arrange your lamps so that you avoid all shadows. Like so. Make sure to take multiple photographs of each specimen, and then you can choose the best one. Otherwise you'll have to go back and do it all over again. And then rinse and repeat. So as well as body shape, you can also utilise geometric morphometrics to look at the variation of other useful features. The lower pharyngeal jaw on the left is part of a set of jaws which sit at the back of the fish's throat and are distinct from the oral jaws. They're thought to likely be modified gill arches and are often specialised within the cichlid family. This specific one is from a Statorochromis alaldi, which exhibits phenotypic plasticity in the pharyngeal apparatus. So this phenotype is adapted to eating hard foods such as snails, and you can see it's really molarised with large teeth and a bulky shape. The gill rakers on the right are on the opposite side of the gill arch, the gill filaments, so you can see that's just a dissected out gill arch. And they also vary based on the diet, so these are quite bulky and short, mirroring the composition of the LPJ. So you can also take photographs of these and assess the variability throughout and between populations using geometric morphometrics.
So, welcome to part 2 of our Geometric Morphometrics video series. In part 1 you learned how to obtain the photographs, and now I'm going to show you how to edit them. So editing the photographs is not a fundamental prerequisite for geometric morphometrics, but it can be very useful for viewing the features when line marking, which can be quite a monotonous task in itself. So anything that speeds that along is useful. And then for the final paper, these graphics can really make your figures stand out. So I'll go through this, but you can go straight to part 3, the landmarking section, if you wish. So I find that GIMP, GNU Image Manipulation Program, is the best software for this. It's free and it has all the required features. After selecting the best photograph of each specimen, I'll show you how to import it into GIMP, how to remove objects such as parts of the pins, contrast and brightness adjustments to better display the features, and then how to cut the photograph from the background, which is really useful for your final paper. So first, locate your photograph. open with GIMP. So now that we've got the photo open, one of the first things you could do is, if your image is a bit dark, you can adjust the brightness and the contrast. So go colors, and then brightness, contrast, and then increase the brightness, which can make some of the features faded, so then you'll need to equally adjust the contrast for that. And then this image has quite a yellow tinge from the lamps that we used. You can make a choice here. You can either carry it on with coloured image, or you could desaturate it. So if you choose to have it coloured, the best thing to do is go colours and then colour balance, and then we can sort of de-yellow that a bit, make it more purple to adjust for it, to make it a bit more natural. But then equally you could also make it monochrome, because the current colours, the sort of brown tinge from being in the ethanol doesn't actually look like what the fish would have originally looked like. So you can go colours and go to hue, saturation, and then just make the whole image grey. But that is completely up to you. Then if you want to sort of remove all the excess and just have the fish as you want it, select what you want to be cropped, and then go image, and then crop to selection, and it will crop it into that box. So this is all that you would really need for the geometric morphometrics. But if you want this photograph to be in your final paper, perhaps on a scatter graph where you're showing your results of your geometric morphometrics, then it would look quite nice to remove the background. So that's the next step. So you click on this lasso tool here, which is free select, zoom in, and then it's really your choice as to how detailed you want it, whether you want it to be absolutely perfect or a bit more rough. So you'll go around the entire fish. A note is, if you make a mistake, like so, don't press escape, because it will undo everything you've done, which will be really frustrating when you get to the end. Make sure you press backspace instead, because you see you press escape. That's all undone now. So now when we get to the end, you just link up with that first point, and then you press this button here, the move tool, edit, copy, and then you can just get a Word document. And then you can just paste that straight in. You can save as picture or do whatever you like with it. The only other thing that you might possibly want to do is, if you have, say, perhaps something there that you don't actually want, quite often you can have a black speck such as these little tiny things here, then you can use the clone tool to remove them. So you click on the clone tool, which is there, and then you hold Control to select what you want to cover it with, and then you just cover it with that. Can look quite messy, but as long as it looks better than what was actually there, then it's fine. And that's all you really need to do. So welcome to part 3. We're going to go through the main part of Geometric Morphometrics, which is the landmarking. I'll be going through how to build the file from the photographs, how to landmark, add the scale and the ID. I'm also going to go through how to do curve landmarking. So normal landmark-based geometric morphometrics has limitations in how it represents the body form because it relies on a limited number of discrete points. And some of the most interesting biological structures actually lack discrete points, so such as the curve of the head, most of the lower pharyngeal jawbone, and the top of the gill raker. So large regions of the object can be unsampled, and then the shape between the landmarks will also be undefined. And such a large loss of morphological information is bad, particularly for the comparative studies that most of you will be doing. The use of more pseudo-landmarks can help this. So like before, like the landmark that's equally between 1 and 5, for example. But semi-landmarks or curve landmarking are another useful method to combat this. So these define outlines and can be used to represent margins of bones, fins, the head, any feature really that would be poorly represented by true landmarks. So once I've gone through how to do normal landmarking, I'll also go through how to do this method, and then you can decide what's most appropriate for your project. So the first step before you do anything else is to make sure that you have some sort of template, such as this one that I made in GIMP, just showing the location and order of the landmarks that you've chosen. This is quite a good example. You can see that I've omitted the tail landmarks there, because generally most of the samples that I've had have had at least one group that's had sort of bent or warped tails from the jar effect. You might get lucky and you might have a nice sample, so you'd be able to include these, but I'll be omitting them. You've got, the posterior and anterior insertion of the dorsal and anal fins, which are examples of true landmarks. And then in the eye, you've got examples of pseudo landmarks. And then say you chose to have a landmark between 6 and 7, then that would be an example of a semi-landmark. So make sure that you have this for when you do your landmarking. So we'll be using TPS Utility and TPS Dig. So if you open up TPS Utility first, select Operation, build TPS file from images, input, and then you select yours. Just select, so you're in the folder with all the photographs you want to use, just select the first image. And then for your output, then go Setup, and then here you can remove any images that you don't actually want to include. And then click Create, and that's now been created. So you can see here you've just got basically a template. You've got no landmarks or no scale yet. You've just got your image names. So the next step is to open up TPS Dig. File, Input Source, and then select that TPS file that you've got. And then you'll find that all your images are now in here. So one of the best things to do first is to set the scale for each image. So you'll go on Image Tools, Measure, and then select your reference length. So this can be anything, but it's easiest to use a centimetre if you're using just a normal ruler. And then Set Scale. And then you'll be able to select what represents that on your image. And then go back to Image Tools, OK, and then here you'll have your scale factor. So you'll go through and do that for all of your images. So the next step, once you've set the scale for all your images, is to give them some sort of ID. This will really help you because if you see here, all of them are just numbered, and they'll just be numbered 1, 2, 3, 4, 5, 6 with no sort of way of actually telling them apart. So you can get this off the image name down here, or if you've just included it in the photograph. So now that you've added the IDs and you've scaled your photographs, you're now ready to start the actual landmarking. So the default landmark cursor is this one, which isn't that great really, and it can be quite hard to see exactly what you're pointing at because it's quite obscured. So I normally change it to this one, and it can be much easier. So if you still need to use your image, you can use that here to see what order your landmarks are going to be in. So also in the default you don't have numbers on your landmarks, so make sure that you do label landmarks and you'll get these numbers which make it really easy to see if you've done them in the correct order or not. So then you just repeat this process for every specimen. So you see, if you do the incorrect number of landmarks, you will get an error message, which is quite useful because it means if you just quickly go through them before you finish it, you don't end up having errors when you actually go through the analysis. So then, if we just zoom in, you'll be able to see exactly where your error is. You can see here the last two eye landmarks are not there. So once you've done all the normal landmarks, if there are any other features that you want to explore, you can then use curve landmarking. So just make sure that you either do all the normal landmarks first and then the curves, or do the curves and then the normal, and don't mix it up. So use this yellow pen here, draw background curves. You've also got to make sure you always do it in the same direction. So I'm going to look at the head curve. So we'll zoom in, and then we'll go up the head. Oops, the daisy. Yeah, if you want to delete anything, just use this on here, and you can use that to delete normal landmarks too. And then you can do it just like that, going along. And then you want to make sure that they have the same amount of curve points. So for that, you can select the curve, and you could resample it and then select the amount of points that you want. You can also readjust where the points are like that. And you could also add points to the curve or remove them. Oops. Like that. So you need to make sure that you redo this for every other specimen and do it in the same order each time. So let's delete those curve points there. And then we'll check how many points are on this one. So there are 13 on this one, so we'll make sure there are 13 on all the others. So then we'll need to resample this one because it's got more points than the other one. And that now has the same amount of points as the other one. So now that we've done all these landmarks and we've saved this, we open it up again. You can check if there are any errors along here as well. You can see they've all got one curve and 13 points and a scale. So if you're going into Morpho.j, it won't be able to read the curves. You have to use TPS Utility again. And this time you want to select append TPS curve to landmarks. And then we'll use this as our input file. Output example. And then create. And now that file that we've just produced there will be able to, this one, will be able to be imported into Morpho.j. Another feature that I forgot to mention that you can use TPS Dig for is linear measurements and angles. So you can use this tool to make angle measurements. Say I wanted to measure the angle of the mouth relative to the lateral line. I could just do this and it will just display it there. And then you can use this to do linear measurements.
So you could use this when you're doing your initial tests to determine how much error each photographing method has. So yeah, just some more features that you can use within this program. So welcome to part 4, where I'm going to show you how to analyse your landmark coordinates that you produced. I'm going to show you how to import the data, how to do a Procras superimposition, which I explained in the introduction, how to remove data outliers and exclude observations, a covariance matrix, how to do a principal components analysis to show the major shape variation, how to represent the mean shape and shape variation in wireframes, transformation grids, and lollipop diagrams, and how to do a canonical variate analysis or a discriminant analysis, which is a method used to find shape features which maximize the separation between groups, and also to see how distinct the groups really are. Open up Morpho.j. So then we're going to go File, Create New Dataset. Make sure that when you import your data, you pick TPS, because if you have text, you'll come up with an error message. Then find your data. So then you'll have your TPS file that you created in TPS dig. So create dataset. And then you've got your new data there. So now you're going to do the Procras superimposition. So preliminaries, new Procras fit, keep it on a line by principal axis, and then perform Procras fit. So the large dots represent your mean landmark positions, and then the small dots represent your Procras residuals. So you can now check if there's any data outliers. So click preliminaries and then find outliers. So the blue dots here show the average shape, and then if you click on any of the cases, the red represents how far they deviate from this average. And if you have one that's particularly deviated, then you can remove it using this. So they all seem pretty similar, so I'm going to keep them the same. You could also exclude observations. So what I will do here is go on preliminaries, include or exclude observations, and then you can select ones you want to exclude. So you want to exclude all the females, all the males, or all of the specific species, or something like that. So before we do the principal component analysis, we need to do generate a covariance matrix, just using this. Select the Procras coordinates and execute. And now we will be able to do the principal component analysis. But before we do that, it's good to create a wireframe, which is a way of visualizing the results. You don't need to do this, but it will be a nice way of presenting your data. So all you have to do is link the landmarks and it will create a link between them. And generally you'll do this along biological features, but it's entirely up to you on which ones you think will look the best. So that's my wireframe template done. It can be quite a laborious task, but once you've done it, you only need to do it once. You can see I've represented the overall body shape. I've got the pectoral fin origin there, the operculum, and the eye, which I think is a good way of visualizing it all. So you just click accept, and that is now saved. So something else that's quite useful to do and will be helpful later is to add classifiers. So if you have quite a small data set, like anything under like 100, you could just
edit them and just add them yourself. So add classifier, and then say species, sex, things like that. But if you have a large one, then this would be quite tiresome. So the best thing to do is create your own classifier file. So I did that, which is here. And you can see I've just done species, location, and region. And then you just do that in Excel, making sure the ID is the same as the ones that you've used in during your landmarking. And you do file, save as, and then comma delimited file. And then you'll end up with that one there. And then what you can do is file, import classifier variables, and then find that classifier file. Open. And then if I go on here and I edit classifiers, you can see that it's now filled them all in, so I don't need to do that manually, which is great. So to go back to our Procras coordinates, we did the covariance matrix, and that enables us to now do a principal components analysis. And that will produce a few different graphics. So we've got the lollipop diagram here. We've got the eigenvalues, which show us which principal component is responsible for most of the variance in body shape. And you can see that the first one is substantially more important than the others. And then you can see the principal components plotted on a graph, the first and the second. So on this first one here, the lollipop graph, you can change that. So you can change it to the wireframe that you created there, which is a lot nicer to look at. You can also view it as a transformation grid. And you can also change any of these shape graphs. So if we want to go on the wireframe, we want to maybe change the target shape to a different colour, and then the starting shape to a different colour, and then maybe get rid of the landmark numbers. So then when I change it back to wireframe, it will have all that now. So it really depends on how you want to do it. You can also change it so it's not on the first principal component, but normally you would, because it does explain most of the variance. Also, just to show what this actually shows, if I just go back to the lollipop graph, the dot shows the mean shape, and then the line shows basically the shape variation. So if you want to go into more sort of detailed analysis, I'm going to open another one which has a bit more data. So I'll just save this. So I'm going to open this data set here, which is a much larger data set. So if I just show you the graphs, this is actually a bat mandible. But it works entirely the same as the fish body shape. And just because this has such a large data set, I think it's a better one to show. So if you remember the PC diagram from before, component 1 and 2. So if you have set groups and the classifiers that you created, you can then go on here, confidence ellipsis, draw ellipsis, and then you can use any of those classifiers to group them and to draw an ellipsis on. So say I want to group by species, and I'll use this to decide on the colours. And now you can see each species, or genus here, is coloured differently, which is great. So then we go to comparison, canonical variate analysis. We give it a name, and then we pick what data set we want it to be on. So the whole bat one, we pick what we want to do on the Procras coordinates, and then how we're going to want to compare the groups. So by species, permutation test, pairwise distances, we can get a p-value, and then execute. So you can see these scores there. And then if we go on results, got all our results here. So here you've got your groups and your observations. Then you've got the variation among groups, so how much variance there is, and then most importantly you've got your p-values here, so you can see whether the groups significantly vary. If we go back to the graphics, so if you go on the shape changes, that shows the shape change associated with the axes of the canonical variant, which is the number of axes is the number of species minus 1. You've also got the CV scores, which is just the scatter plot of the CV scores. We can also do, because this data has longitude and latitude, you could do a regression. So if I go regression, then you can pick your dependent or independent variables. So say for our dependent, we have all the bats and we have the Procras coordinates. You select that like that, and then you can use your covariate latitude, perform a permutation test, you can do a pooled regression of species, execute. So then when we go on our results, you can see the p-value is less than 0.05, so it is significant. And doing this, you can see any sort of association between any of your covariates. So if you go on here, you can see it did say there was an association. You can see that there is a slight association between it. You can also, again, use your confidence ellipses here based on species. Oh, sorry, make sure to colour them. And you can see it's the way it goes up. And then I'll show you how to export your data set. So you just select the data set that you want to export, or data set, and you can pick what you want. So normally you would want your principal components. I'm just going to export that. There we go. You can also select any of the classifiers you want to export, and then just save it as what you want. So welcome to part 5, SPSS. I'm going to show you how to import your data from Morpho.j, how to run manovers, make PCA diagrams, and do discriminant and cluster analysis. So the first thing we're going to do is open up Excel. And what you want to do is you want to open the file that you saved with your PC scores. I'll just find out which one this is. And then it'll come with this, but just press finish. And so you can see we've got our IDs, we've got all our classifiers, I've got the centroid sizes here, and then all the principal component scores. So I've got another one which is a little bit more simplified that I'm just going to use for the example. And then I have species numbers here, which is something to add in if you want to do discriminant analysis, because you need numbers rather than string variables. So just assign each of your species, or if you're grouping by something else, each of your groups a number, because that'll be really useful later. So then we're going to open up SPSS. New data set, and then we'll go back to our data. So select all the data, but don't do the column names, because otherwise it will think they're all string variables. Another thing is you'll have a lot of principal component scores from your analysis, but I normally only select the first 10 because, as you'll have seen from the loadings on our eigenvalues, that they describe most of the variance, so the others don't actually really show anything. And when you're copying into SPSS, if you have massive data sets, it can crash. So there's no need for any further than 10 principal components, really. So now we've got all of our data in there, but then you'll need to name your columns. So you go into variable view, and then I'll just copy them off here. So var ID, species. You also can't have spaces in your variable names, otherwise it will come up with an error. So when you go back onto data view, you can see that all of our columns are now named. So now we can start doing some analysis. The first sort of simple one to do is the MANOVA, so general linear model, multivariate, because we've got more than one. And then in your dependent variables, just put whatever you want to check if there's a significant difference in this variable between the groups. So I'll select all of them. And then in fixed factor, put your species. And then we want to compare between the groups. So we'll do a 2-key post hoc. You want to get some stats results so we can actually see what's going on. Just have a look at that. So this is sort of just a prerequisite to the more complex analysis. So you can see here we've got our sample sizes, then we've got our multivariate tests. So you're looking at this one, Wilks lambda, and if this is lower than 0.05, then you know that as a whole there's a significant difference. But if you want to look at each of them individually, go down to species, and then for which of these have the same low p-value, you know that there is a significant difference between the species. So you can see it's in most of them, centroid size and log centroid size, PC1, 3, 4, 5, etc. So what we can do now is the post hoc we did the multi comparisons now compares it between each group. So you can see, say, we've got just 3 species here just to keep it simple. So Blower versus Calyptera is significant for centroid size, etc. So this can be quite useful. But really, you want to find out how significant each of these variables are. So for that, we will be doing a discriminant analysis. So I am going to go to Analyze, Classify, and then Discriminant. And then you insert the group numbers. So those are the ones that we had that we assigned to each of our species. So I'll go species number, define range, and then just 1 to 3. And then you select everything you want to discriminate by. So we've got centroid sizes and then all our shape variables into the independence. And then we're going to use stepwise method. Then you can use box M. It's a good test to understand if you're following certain assumptions for the discriminant function analysis or not. Also use Fisher's coefficients, so the loading of the discriminant functions. And then method. And then you're going to want to use the probability of F, 0.05, 0.1. This means in each variable, the software is running a kind of ANOVA, and if the p-value for a certain variable is more than 0.05, and less than 0.1, then it's retained. If not, it's not considered. And it does this for all variables. So that's the stepwise process. Start with one variable, then the second, then the third, and so on. And if it looks as if the F parameter is increasing, if it does not, then you don't need to add more variables. So the discrimination is maximized. Okay, then we go on to classify. So I normally pick all groups equal, but if you have like particularly different sample sizes, then you'll want to compute from group size. And then within groups in the covariant matrix, which is generally fine if you have a big sample size, otherwise you can use separate groups. Then you can plot the combined groups, so we'll get a nice plot of discriminant function 1 against discriminant function 2. And then in order to have the results, you want to do case-wise results, summary table, and I'll leave one out, classification. You can also choose to save your discriminant scores. These can be used simply to represent the discriminant function later on. So what I'm saying now is find the function to discriminate between these species categories based on the selection of variables you think is adequate enough to discriminate between these groups. So in this case, we've included centroid size and the principal component scores. So it's going to load our results now. Okay, so this is the first thing that you really want to look at, Box's test of equality. So generally you want this to be insignificant, so that is, which is good. But even if it isn't, as long as your log determinants are pretty much equal, then you're okay. So then you can see the stepwise statistics, what's happening. So the software has done each of these steps, and it's selected principal component 3 first. So the software thinks that this is the most important variable. So these are the variables that were selected to be the discriminant function vectors, and it's showing you the steps it managed to do. You can see the more steps it's doing, the more the Wilks lambda is decreasing. Wilks lambda, sorry, is just decreasing. So these are the variables that were not entered in the analysis, so it doesn't think they're important. And then these are the stats that you might think about putting in your final paper. You can see it's created 2 functions, which no matter what amount of variables you do, it will only do 2 if you do 3 groups. And then you can see for the first function, it's contributing 85.6% of the variance, and then for the second, 14.4%. So this table at the bottom is the part that people are generally bothered about. So ignore the original, just look at the cross-validated correct classification percentage. You can see that these are all very high. Sometimes you do it and you just have like something very low, like 30 or 40 or sometimes even 0. So this shows strong discrimination between the groups. And then you can also see in this plot here, that's represented here, there's quite, especially group 3, which is Calyptra, it's completely separate from the other 2 clusters, and even though there is overlap between clusters 1 and 2, which is below a time of 10 ASI, there is definitely some discrimination between them, so that matches up with these results. So you would report these in your paper. So you can kind of choose what you want to do with discriminant analysis, and you can kind of do it as an experimental approach if you like. So I'm going to show you now discriminant analysis, but this time I'm going to include some other variables. So let's find this. So here I have just normal classical caliper-based measurements for each of those. So I'm just going to copy them and add them to our data. You can also see that we saved the discriminant scores there, which could be useful later. So now we also need to name all of these. Okay, So now that I've put those in, we can include them in the same analysis and see if any of them are really contributing to the distinction between the groups. So up to principal component 4, and then put in all of these. Okay, now you can see there that the box diagram is unfortunately significant, but there doesn't seem to be a mass amount of difference between the log determinants, so that's okay. You can see on the stepwise it still thinks that principal component 3 is the most important, but now it has head length and eye diameter as the next most important after that. So it'll be interesting to see how it has discriminated based on that. So if we go to the bottom, there's actually higher distinction than before, which is interesting. You can see that groups 1 and 2, which is below a time and assay, are much more separated than they were. There's less overlap. So this suggests that principal component 3, head length and eye diameter, are really important distinctive features. Something else you can do is if you have unknown, something that's got unknown to unknown species or unknown location or something like that, you could use this to predict where it will be. So if I just, for example, just get a couple of these and make them unknowns, we can then see if the discriminant analysis puts them in the right groups. So press OK. Now we go to that table just before, which is here. It'll have those unknowns, and it's predicted that one to go in group 2, that one to go in group 1. It's predicted all of those to go in group 3. So if we go to the bottom, it has here ungrouped cases, and it predicted 20% of them to go in 1, 20% to go to group 2, and 6% to go to 3. So this can be a really useful way of trying to find out what any of these species actually are, what their real identity is. Lastly, now that we know what the important grouping cluster, we can have a look at how many clusters it thinks there are. So if I go in the categorical variables, I put the first 3 principal components, and then I'm also going to put head length and eye diameter because they seem to be pretty important. Oops. Okay, sorry, in that one. And then you can pick either of these. Get some pivot tables, and we can create a variable for that as well. And then we can see how many clusters it thinks there are. So it thinks there's 3 clusters. And then if we go more deep on this, oops. So we're on model viewer now. You can see the sizes of each cluster as a percentage as well. We can also view the predictor importance. So this is going to say what it thinks is most important. So it thinks, same as the discriminant analysis, the principal component 3 is the most important, and then head length, and then eye diameter. So this is quite important because it's saying the 3 clusters, which are up on the same thing, and we can see that's likely species, but we can actually find that out because we created the cluster membership variable. So if we just do something like a histogram, and then we colour by species. Sorry, the histogram is of species, but then we get cluster membership number there. And let's go to that. And you can see that actually these clusters are not clustering by species, which is quite unexpected, really. But this is a good way of checking it because you would assume that it clustered fully by species, and it actually hasn't, so it must be clustering by something else. Quite often it can be that it's clustering by standard length. So if I was to get standard length, and then try it with that membership. You can see that it does seem to be clustering by standard length, because most of the smaller fish are going into that purple cluster 3, and most of the larger are going into that green cluster. So now we're going to go through analysing your data in R using the Geomorph package. Functions in this package allow you to read, manipulate and digitize landmark data. You can generate shape variables via Procras analysis for points, curves and surface data. You can perform statistical analysis of shape variation and covariation. You can also provide graphical depictions of shapes. And then we can also look at allometry and sex versus species. So the first thing to do is open up R. So the first thing you do in R is to clear your working space. You don't need to do this if you haven't used R before, and you're doing it so any previous code doesn't interfere down the line. Then you need to install the required package Geomorph and then load R. So install Geomorph. So now we're going to load that package. Then we can set our working directory, or you can just write it in to your code. So next thing you want to do is import your landmark file and give it a name. So you can see it's come with no curve detected because the example I've used doesn't have any curved landmarks, and it's coming correctly. So if I wanted to show the data that I've brought in there, it's just got all my landmarks there. So the next thing I want to do is import some classifiers. You can just add in a classifier file, but there can be problems with it, so often the simplest solution is the best. So you just create a file such as this one. And here with each of the IDs that will correspond with your landmarking, I've got locality, year, species, latitude, and longitude, but you can't just import it like this. So the best thing to do is just copy all of that and then transpose it like so. And then you can use the ampersand function to create a list like so, and then you'll be able to import that in using species factor. So just to show the example, so I've got locality factor, and then in there I'm going to paste in everything I've got there, taking away the empty ones. And then I just enter like so, and then I can check if that is a factor. True, so it is a factor. And then you do this for all of the classifiers that you're interested in. So once you've added in all these classifiers, the next step is to do the generalized Procras analysis. So create a new name for these new coordinates that you're doing and use that function. So now we could do plot our original coordinates, just there, and then that's what you get. But if we plot these new coordinates, we get that. We can also run a principal components analysis. And then we can plot this. You could also colour it by species or anything else that you're interested in, like so. You can do a summary, so you can see the eigenvalues, see how much the portion of the variance each component is responsible for. Right there, so you've got 23.6%, 13.6%, etc. Now, for my fish file, I don't have enough female specimens to show you how to use species versus sex and potential sexual dimorphism in body shape, so I'm going to use some other data as an example. And it's exactly the same process, but these are bat mandibles rather than entire fish specimens. So I've read in that new data, and then I'm going to do the same as before and add all my classifiers.
Here I make a little mistake where it's going to read in latitude and longitude as factors instead of vectors, so they shouldn't have the quotation marks. So if you're doing the same, make sure not to include those.
Okay, so I've added my data and I've read in all my classifiers. I need to also do a progressive analysis on this data. Okay, so before, if I was to just plot that to show you what it looks like. See the general shape and all the different individuals. So next we're going to create a data frame from the shape data using sex and species classifiers, so we can explore everything a bit more in depth. So in order to do the following functions, we need to use this data frame. And we'll look at the relationship between centroid size and shape variation of this example, the bat mandible. So we're going to use this function to perform the process ANOVAs to assess our statistical hypothesis looking at shape variation and covariates. So give this a name. Then we're going to look at our coordinates. And then using the summary function, we can see if there is a significant relationship between size and shape. And you can see it appears that there is a significant association between shape and size. So to visualize this relationship, there are many functions. We could use the plot function with a regression type, or we could use plot allometry, and we'd end up getting the same graph. We could use prediction lines, which are first principal component scores of fitted values from the PROC-DLM, or we could use regression scores, which are standardized projected shape scores along the axis defined by the regression of shape or size. So and we will get this graph here with the predictor line. We can also, if we like, colour this by species. Like so. Then we could also use the regression scores instead, by just using this different method, like so. So if we were to use this instead and not use the allometry model, and you can see we'll get the same graph there. So an important detail with these plots is that the predictor line and the regression score are model-based projections of shape data, and changing the model will change the outcome of the plot. We could also perform a two-block partial least squares analysis to find the correlation between shape and size, which is not based on a particular model. You can see that there does seem to be significance, and if I plot that as well. So we can also do a plot of the common allometric component, which will always be the same as the PLS plot, irrespective of the type of shape allometry model. So just colour that as well. And you can also colour this by other things as well. So we could colour by
6 if you wanted. We can also append a size vector to a matrix of shape variables and perform principal components analysis called a size shape PCA. Like so. So those are really good ways of testing allometry and presenting those results. So we can also do some more complex allometry analyses. And then we can do a summary of this, like so. We look at the influence of sex and centroid size and sex species. So what we can do is we can use the function pairwise and do a pairwise test which can be used to calculate the pairwise comparisons between group levels. So I do so you can see the pairwise distances between the means. We could also do this. We can do the same for species, not just sex. Something else we can explore is morphological disparity. Go this for the entire data set coming to allometry without covariates, and basically we'll end up getting cross variances and some tests of significance, etc. So if I was to do this for the entire data set, accounting for allometry, then I can do without covariates using the overall mean.
So you can see with these results here, we've got the cross variances for each of the groups. So we've got this species, the females of this species, the males of this species, and the unknown sexes of this species, etc. You can see the pairwise absolute differences between each of these groups. And then you can see the significance of these differences. So, for example, female P. auritus have significantly different distances between the percustance variances than female Dogontoni. So you can do this for all your groups and get some really interesting results. So now I'm going to go through some nice ways that you can visualize your shape data. We can estimate the mean shape of our set of aligned specimens, or consensus shape, using shape. Oops. And now you can see we've got all the coordinates there, but we really want to visualize that so we can plot it. So if I call it consensus, okay, and then we can plot that with the axes that we want. Like so. And now we want to plot all of those points. Okay, so you can see that we've got all of our points plotted, but then we want to put the mean on top as well, so our consensus shape. And then colour it whatever colour you like, and do it red. Oopsie. Any slight little mistake. There we go. So now, if I just zoom into that, we've got all of our points plotted and then our mean shape in red on top, or our consensus shape. We could also do a transformation grid. So, oops. So if you look at the previous literature, you'll see a lot of plot tangent space and things like that, and they're now defunct. So the stuff I'm showing you now is what they basically got replaced with. It's a better way of doing it, basically. Okay. Okay, so this is the transformation grid, which is another way of visualizing the mean shape with the shape change from the sample. But something else that is really cool, or can be really cool, is to do individual deformation grids for basically any part of the graph that you're looking at you want. So I'm going to do it as an example with principal components analysis, with principal components score 1 against 2 on the x and y axis, but technically you could really do it on any graph that you've used. So I'll show you this one first. Oops. Just give that a name. And then we use this function, pick and plot, which is really cool because what it enables us to do is, so here's our principal component graph, PC1 against 2, and you can see all your points there, and basically you can pick any point you like in this graph, and then do the deformation grid for it. So if I just pick that part, it says, sorry, and it'll appear, which is just here. And then I can say whether I want to save it. So say I've picked, for example, just one in the middle of that first species, and then you could do a deformation grid for just a representative specimen of each, or point of each species. So then I can pick another point if I like, and then I just pick one from, say, one of the other clusters, and then I can save that as well. So you can make some really cool figures from this, because you could have that in your paper and then have representative deformation grids, which can be a lot more exciting than just looking at principal component scatter plot. But you don't have to do that sort of plot. You could also do, say, if you watch the allometric part, you could do one of those. So if I'm just going to do this as an example. So let's clear this first. It gets a bit busy otherwise. And exactly the same thing we did before on the plot allometry. I'm going to use the predictor line, but you could do this with any of your graphs, the regression scores, or whatever method you want. And then again, we use this pick and plot function. This one. So we've got our predictor line like before, and I can just pick one. So this is quite cool with the allometric model because we can go for one right at the top. And let me just that one, and then we could save that. And then we could pick another point from right in the bottom. And then we can just keep doing that. So yeah, it's a really cool function and should hopefully help you to display your results really nicely in your paper.